A quick introduction to metagenomics. Now, what on earth would we be interested in metagenomics for if we're, for example, clinical microbiologists? And one of the issues is if we look at the way in which we do diagnostic microbiology, we're stuck here in the 21st century with 21st century problems, but we're still using 19th century techniques. So these are two key papers in the history of microbiology. There's Christian Graham invented the Gram stain back in 1884 and published that. And Robert Koch in 1881 was able to grow bacteria on solid media for the first time. And we've relied on those two approaches of you looking down a microscope and growing things on solid media ever since then, for well over 100 years. Could we not do better? So a few years ago I got involved in a study of the sugar toxigenic E. coli outbreak that struck Germany in 2011. And what we wondered was whether we could actually get the same information from the fecal samples we had from the outbreak as we got from culturing them, as our German collaborators got from culturing them and culturing the E. coli from them. Could we get that same information just by doing sequencing without culturing first? So the idea was, in the initial studies, they had cultured the E. coli from the stool samples. My computer's making a funny noise again. Oh, it's just stopped. You get the E. coli out and then you sequence the E. coli. Could you not get that same information directly by sequencing the stool sample? So, in essence, what we're doing here is we're taking a complex mixture of lots of different microbes in the sample. We're extracting DNA from all of them mixed together, and we sequence all that DNA, and then we do the hard work, the kind of heavy lifting, is all done in the phase of bioinformatics, where we're trying to analyze those sequences and do taxonomic assignments for all those sequences, and then use that information to reconstruct what organisms were present in the original sample, what taxonomic phylogenetic group they belong to, but also what other interesting features of their biology we might be able to reconstruct from these sequences. In fact, we weren't the first to do it. Others have tried this. I think I've mentioned some of these, one of these at least in a previous talk. But there's been a number, even with an African focus, there's been a number of examples where people have used just sequence-based approaches to discover new pathogens that are causing disease and are otherwise hard to diagnose, hard to recognise what's happening. So that's a little bit about diagnostic metagenomics. Worth also saying with diagnostic metagenomics, it has a very open-ended feel to it because you're just sequencing DNA. You don't know what you're going to find. You could obviously make PCRs that for each specific pathogen, but what happens if the pathogen that's actually there isn't one you've got in your repertoire? With metagenomics, you can actually still find it. So there's one interest in metagenomics as a diagnostic approach, but there is another interest in this, and that there's been a massive interest in recent years in the human microbiome. So, we all got excited about the Human Genome Project, and it's hogged the limelight. But in fact, our human genome is relatively simple when you compare it to the accumulated genomes of all the microbes that also live on and in our bodies. So this complex community of microbes, known as the human microbiome or the human microbiota, it's much more complex in terms of the numbers of genes and in terms of the metabolic repertoire encoded in those genes than what we carry in our nuclear genome. This is one of the most complex known microbial communities, and probably around 1,000 different species in there. Depends from one individual to another, but somewhere between 500 and 1,000. You can argue, in any one of us, there are 100 times more bacterial cells than there are stars in our galaxy. And if you take all the humans across the whole planet, we are carrying around more bacterial cells than there are stars in the known universe. So this is a huge community. People argue that you often hear this thing that there's 10 times as many bacterial cells as there are human cells in you. That is perhaps a bit of an exaggeration. It depends what you call a cell. If you call red blood cells, some people say they're not cells because they've got a nucleus, they're corpuscles. If you call those cells, then we've got about 30 times 10 to the 12 human cells. If you exclude the red cells, then we've got 5. But either way, there are still many more bacteria, 38 times 10 to 12 bacteria is the estimate, than there are human cells. And if we exclude the red cells, then, not quite 10 to 1, but something like 8 to 1 in terms of favourite bacteria. And particularly the gut microbiome, which is the most abundant and complex microbiome, there are many positive aspects to its function. It provides protective functions, prevents pathogens from gaining a foothold. And we know, for example, if you get given broad-spectrum antibiotics, you become much more susceptible when your gut microbiota has been disrupted to infection with things like Clostridium difficile. So we know that this microbiota has protective effects. There are structural functions as well, and if you don't have a gut microbiota, you don't develop your immune system properly. So notobiotic mice, where they keep them and propagate them without any microbes around, their gut immune system is not normal, and even the gut development in terms of the histology of the gut is not quite normal. So it's needed to, if you like, train the gut and the immune system to behave in the right way. And then there are many metabolic functions of this microbial community as well. It basically allows us to digest things that we otherwise wouldn't be able to digest. And so a sizable fraction of the calories that you take in may have been recycled through or cycled through the gut microbiota. One of the key features of research in this area in the last few years, though, is not just recognizing that this thing exists and it's interesting in its kind of general sense, and not interesting, and not just because bacteria can cause disease in the form of infection, but it's now becoming clear that this microbial community can influence the balance between health and disease in many conditions that you wouldn't ever think of as being infectious conditions. So this was one of the key landmark papers that kind of kicked the whole thing off, where they showed that the gut microbiota in mice, was it mice or rats? Yeah, mice, was actually able to influence the weight gain in those mice. So they had mice that were obese, genetically determined to become obese. They developed a gut microbiota in those mice and then transplanted it into new mice, completely new mice. And then they also took their lean litter mates, took their microbiota, and what they were able to show in transmitting the microbiota was that they could transmit the propensity to put on weight or not. And there's been a great deal of interest in kind of weight homeostasis and the influence of the microbiota on that. But there are many other conditions now. So inflammatory bowel disease, people are interested in that. Liver disease and the influence of the gut microbiome. There's even interest now in the way in which your gut microbiota might influence your psychological well-being. And this is a huge area of research. The methodologies we've mentioned before, we've got the microbiota sample, and you can do culturing. There's a guy called Didier Raoul in Marseille who, after it initially became clear that people were interested in these microbial communities, but obsessed with doing it by the sequence-based methods, he pointed out, well, you can culture most of these things if you try hard enough, and you don't have to get obsessed with sequencing. And to ram home the point, he came up with this I've kind of got used to it now, and I started to like it, but I hated it. It's got this neologism of culturomics, you know. So you've got genomics, metagenomics, and culturomics. All it means is old-fashioned microbiology, where you just grow things. Although he does have a lot of tricks where you can do it fast. You can have lots of different conditions, lots of different atmospheres, lots of different media, and so forth. Then you can do metagenomics, where you extract the DNA and sequence it, and you can start making functional inferences, as I mentioned before. You can do the taxonomic analysis. You can do that with 16S as well. If you're doing culture, you can also then do whole genome sequencing. And there's a lot of interest in, can you get the same information out of metagenomes as you could by culturing and then sequencing? How well can you actually separate out the different sequences and bin them into taxonomic bins that you can say that definitely is part of that genome of that organism? There's a lot of interest in that. One of the hurdles is that at the moment we're still relying on short read sequencing, so that makes it quite difficult. I think, you know, given the progress that Abdul pointed out this morning, once nanopore and long read sequencing gets to take off and be used in this, it will become a whole lot easier to reconstruct genomes of individual organisms from a metagenome, because you'll have that high contiguity. Here's another figure that just shows the same kind of things that you can do in terms of workflows. So you do your initial quality control, you're doing functional assignments, assembly, taxonomic classifications, and you can get the metabolic profile, you can get new genomes, you get taxonomic profiles. And one phrase that's commonly used, this term that's commonly used, is this idea of binning, that you basically have a load of, you'll end up with loads and loads of contigs when you've done your assembly, and you want to say, well, those ones belong to E. coli, those ones belong to Bacteroides. And you can do that by using taxonomic assignments. You can say, well, the gene is clearly 99% or 100% identical to the same gene in a reference strain of E. coli. But there's a lot of interest in smart methods to actually say, well, genes that may be not seen in any other E. coli but still belong to your E. coli, can you bin them with the rest of the genome? One of the things that is commonly done is to perform a quick taxonomic assignment using an approach that uses what we call K-mers. So if you do homology searches with all of the sequences you've got, you can do homology assignments and use homology to do taxonomic assignments, but it's extremely computer intensive and extremely time consuming. So a quick approach that most people use is that they use this approach based on K-mer. So K-mer is means effectively words or strings of sequence of a known length, of a particularly defined length. So if we said the length was 12, we'd call it a 12-mer. And there would be 12, we look for all the strings, all the words of sequence, if you like, that are 12 characters long. And if you look at sequences in that way, just like if we were trying to work out, oh, is this, I've got some text in front of me, is this text in English or in Wolof? And one way would be just to count the, say, strings of 3 letters. And if it was English, you'd see THE as a very, very abundant string, and you'd see AND as a very abundant string. And in Wolof, you'd see a different pattern. So it's the same kind of approach that we're basically looking to look at these patterns of words to identify what's in front of us and classify it. And these approaches are actually quite sophisticated because they rely on libraries of K-mers that have been pre-computed to map onto the reference genomes that are in the reference databases, the NCBI databases, and make assignments down a taxonomic hierarchy. So it'll start off by saying these reads can be assigned to bacteria rather than, say, humans or plants. Then it will say these can be assigned to the gamma proteobacteria. These ones can be assigned to Enterobacteriaceae. These ones can be assigned all the way down to E. coli. And so it does some quite sophisticated mapping. And you'll be seeing some of that later. that's just a brief introduction to point towards the technologies that you're going to be using. I can't leave this subject without pointing out that although I'm an enthusiast, I would say I'm a skeptical enthusiast about microbiomes. And there's this guy, Jonathan Eisen here, who is also in the same mind. He likes doing this kind of research, but he does not like the way in which it gets oversold and people exaggerate and overinterpret the results of microbiome research. He calls it microbiome mania. And he has a website where he maintains a list of people who committed this crime of overselling. And we do get these silly ideas that, you know, down there, that cartoon, that basically your microbiome is controlling you. And you can say to your wife or your husband when you start putting on weight, it's not me that makes me go to get the cake out of the freezer. It's my microbiome, don't blame me. You know, you can go down that line if you want to. There's just that figure there just shows you some of the conditions which people are trying to implicate microbiomes as having a causal role in the pathogenesis of disease. And if you're interested in this, there's a guy, Ed Yong, in the UK, just about a year ago, wrote a book called I Contain Multitudes, which is quite a nice introduction to this whole microbiome stuff. It's also worth stressing that there's even a kind of slightly sinister side to this. There's been this idea that if you have a caesarean section, if a woman gives birth via caesarean section, the microbes that are normally in the vagina don't meet the baby. The baby doesn't get normal microbiome, and this could have bad effects on the baby. We do know that there are some things that are more common in babies people that have been delivered by caesarean sections. Asthma, for example, is more common in babies delivered by caesarean section. But it's not clear, you know, what the causal links are and the mechanisms. But some people have taken upon themselves that if they have a baby delivered by caesarean section, they take a swab from the mother's vagina and rub it all over the baby's face as a way of saying, yeah, you are going to get this microbiota whether you like it or not. And this has become one of those things that is extremely fashionable and almost dogmatic. And I was speaking to an American microbiome expert, Julie Segre, and she was saying that, you know, there's almost a stigma now in the US if women have a caesarean section because you're denying your baby the right start in life. And she said the evidence for that is extremely slim, if non-existent. The other thing we get in the UK is that, and in America, is that people have gotten onto the idea that things like Clostridium difficile or other conditions, inflammatory bowel disease, may benefit from a fecal microbiota transplant. So you actually take stool samples from a healthy person and give them to the diseased person. It sounds a really disgusting thing. It is pretty disgusting. And it also sounds quite cranky. And for years, people were doing this. The pioneers, we would say now, were doing this and saying it works. You know, we've done 99 out of 100 people who've got chronic C. diff, and we give them a microbiota transplant, they get better. It's now tipped over to there is now accepted practice for a recurrent C. diff that you can do this, but people are cottoning on and doing it at home and saying, well, maybe I've had a bit of diarrhea for a time and I've got Crohn's disease or something. I'll get my wife to give me a transplant from her feces, and they go and buy all the stuff and do it at home. So it's kind of this stuff gets a little bit out of control. And interesting, you know, there is this idea with a lot of new ideas that they go through this so-called hype cycle where you get some new thing that's discovered and everyone gets really, really excited. This is going to solve everything. And you get this peak of inflated expectations. And then people start to realise, well, it's not solving everything. It doesn't work as people are saying. And they get this trough of disillusionment. And then slowly it gets contextualised. Okay, it works in these conditions, but not these conditions. It works if you do it this way, but not that way. And you end up with a more mature view. And I think it's hard to see where we are on that hype cycle. And I guess it varies depending on what application of microbiomes you're looking at. There's this guy, William Hanage, who's an English guy who lives in the US. And he managed to persuade Nature to publish this little skeptical piece that he wrote, saying that basically microbiome science needs a healthy dose of skepticism, and he pointed out in pre-scientific times when something happened, people didn't understand. They blamed it on spirits. We must resist this urge to do that. If anyone watched it, there was a film called The Martian a few years ago, and the key tagline in The Martian was that he had to work on his own wits to survive on Mars, and he ended up having to learn how to grow potatoes out of his own manure and stuff. And his tagline was, I'm going to have to science the shit out of this to survive. And our challenge with microbiome research is obviously we're going to have to science the shit out of shit. And that's more or less what I was going to say. So, as I say, I'm the warm-up artist. You now get the proper scientist who's going to tell you how to actually do it. Thank you very much.